On behalf of the leads of this initiative and our Network for Direct Patient Engagement implementation team, we really appreciate the opportunity to introduce our participant engagement and cancer genome sequencing network. And we're very excited about today's webinar. The goal of my presentation today is to introduce you to our network. The Blue Ribbon Panel, Recommendation A, called for a network for direct patient engagement. The panel said we should enlist direct patient engagement through a federated network where patients would be offered comprehensive tumor profiling. The panel went on that patients are eager to provide their data and that efforts to reach minority and underserved populations will be a high priority. Our genome characterization efforts, such as the Cancer Genome Atlas or TCGA, have been very successful at providing insights into cancer, but we know that research gaps remain. On this slide is one example of a type of research gap. If you look at the race distribution of tumors in TCGA, only 14% of these tumors came from minority populations. One strategy to address these research gaps is a direct to participant engagement strategy. Therefore, we launched the Participant Engagement and Cancer Genome Sequencing Network, or PE-CGS. The purpose of this network is to promote and support direct engagement of cancer patients and post-treatment cancer survivors as participants in cancer research, and to use such approaches for rigorous cancer genome sequencing programs to address knowledge gaps in genomic characterizations of tumors. These knowledge gaps include rare cancers or rare cancer subsets, highly lethal cancers, cancers with an early age of onset, cancers with high disparities in incidence and mortality, and cancers in understudied populations. We have just recently funded our first 3 groups for this network, 2 research centers and a coordinating center. Each of these research centers will be leading independent participant engagement programs that will aim to address specific gaps in genomic characterization. And we're going to be hearing from one of these research centers today. We also have a coordinating center, which is providing the infrastructure support for the network and facilitating our network collaborations. This network as a whole together will function as a collaborative network, addressing common issues, sharing best practices, and incorporating lessons learned, and using common methods where appropriate. This network will aim to address our research gaps in genomic characterization and to define optimal methods of participant recruitment. And with that, I'm happy to introduce our moderator today, Dr. John Carpton. Dr. John Carpton is a professor and chair of translational genomics and director of the Institute of Translational Genomics at the Keck School of Medicine in USC. Before joining USC, Dr. Carpton was a division director at the Translational Genomics Research Institute, or TGen. Dr. Carpton is an internationally recognized expert in genome science and possesses unique training in multiple disciplines, including germline genetics for disease risk and predisposition, somatic cancer genomics, health disparities research, cell biology, functional genomics, and precision medicine. So I'm going to turn the microphone over to Dr. Carpton, who will introduce our speakers today and our webinar. Thank you.
Thank you, Leah. Hopefully everyone can hear me okay. And of course, I'm honored to have the opportunity to participate in today's Moonshot Seminar. I'm especially excited about today's seminar as it represents, again, the Moonshot study related to the patient engagement in cancer genome characterization U2C network. And today's seminar will be brought to us by 2 outstanding and distinguished scientists who serve as co-PIs for this study. And they're both tremendous and long-standing colleagues of mine, Drs. Cheryl Willman and Jeff Trent. Dr. Willman has served as the director and CEO for the National Cancer Institute designated, or NCI designated, University of New Mexico Comprehensive Cancer Center since 1999. She is a distinguished professor of pathology and medicine at the School of Medicine at UNM, where she holds the Maurice and Marguerite Liberman Distinguished Chair in Cancer Research. Dr. Willman is an internationally recognized physician scientist, a national leader in cancer precision medicine, whose research focuses on genomics, next generation genome characterization and computational technologies to identify novel cancer causing mutations that can be translated to better cancer diagnostics and therapeutics. And she's also been a staunch leader in the field of cancer health disparities. Dr. Trent serves as president and research director for the Translational Genomics Research Institute, or TGen, in Phoenix, Arizona. Prior to forming TGen in 2002, Dr. Trent previously served for 10 years as the scientific director for the National Human Genome Research Institute, or NHGRI, on the NIH campus in Bethesda. Under his guidance, NHGRI's Division of Intramural Research became an internationally recognized cancer research center in human genetics and other diseases. Dr. Trent, of course, is an amazing scientist, outstanding leader and expert in cancer research. He specializes in developing and integrating novel omics technologies, supporting studies of and supporting the molecular studies to identify changes and alterations related to cancer risk and progression. Doctors Willman and Trent will be presenting on their recently funded U2C grant, focusing on discovering the genomic and genetic, environmental, and behavioral mechanisms of cancers that include hepatobiliary, genital urinary, as well as colorectal cancers that disproportionately affect Hispanics as well as American Indians in the American Southwest and Southern Plains, which may underlie their significantly higher cancer incidence and cancer burden and poor outcomes. So I'm really excited, as all of you are, as to hear about this tremendous study so that we can learn more about engaging underrepresented minority communities in cancer genome characterization studies. Dr. Willman.
Cheryl, are you on mute? Cheryl, you're muted at the moment.
Cheryl, can you unmute your mic?
There
Yes,
we go.
can you hear me now?
Yes, thank you.
Well, good morning, everyone. Thank you very much for attending today. We apologize. I know there were some difficulties with the Zoom link, and hopefully if you're here, you've successfully entered the Zoom room. I'm very honored this morning to present our participant engagement Cancer Genome Sequencing Research Center with my MPI, Dr. Jeff Trent of TGen. And as Dr. Carpton said, the title of our center is an engagement of American Indians of Southwest tribal nations in cancer genome sequencing. I don't seem to be able to advance the slides. Can you guys? There we go. I have nothing to disclose, both related to the NCI Moonshot presentation or our PE-CGS center. To start today, particularly in this time when our nation is in such agony over racial and ethnic disparities, social justice, I want to give all of you a little background about American Indian nations. I've been very privileged for the last 35 years to live in the state of New Mexico and to really work closely with tribal communities and their health concerns. But there's really important history that I find a lot of Americans are not knowledgeable of regarding tribal entities. In 1893, the Indigenous people of the United States, through a congressional act called the Dawes Act, began to be forced to disassemble or dissolve their tribal entities. This act began to allow individuals to buy tribal lands that led to a significant loss of reservation land held by many tribal entities. The Dawes Act also established Indian schools. And for those of you who are aware of the impact of that on tribal nations, many tribal communities and tribal families were forced to send at least one child away to a remote Indian school to be Americanized, if you will. This was a tragic disruption of American Indian communities that is still felt today. In 1924, citizenship was actually conferred on American Indians. They were recognized as human beings who deserve the right to have citizenship within the United States. And effectively through this act, they could become dual citizens of both their tribal sovereign entity and the United States itself. But interestingly, it wasn't until 1948 that American Indians gained the right to vote. And I'm very proud to show you this picture in this slide of Miguel Trujillo of the Isla de Pueblo. Miguel Trujillo was an educator, a teacher in New Mexico who went off to World War II and served in the Marines. And when he returned in 1948, he was not able to vote. And he began to fight for American Indians to have the right to vote and succeeded in a suit called Miguel Trujillo versus Garley. At the same time, another suit was proceeding in Arizona, Harrison versus Levine. And both of these lawsuits were successful, allowing American Indians for the first time to have the right to vote in the US. There's an interesting backstory here. Both of these trials were led by Felix Cohen, who at that time had really written the book of the federal book on Indian law and was an amazing civil rights attorney that came to the Southwest and accepted these two cases in collaboration with Mr. Trujillo and Mr. Harrison to take these cases forward. I've given a couple references in this slide, which you'll be able to access later, which has amazing history regarding these important civil rights trials and their impact on Native communities. It wasn't until 1962 that the Navajo Nation got the similar right to vote through a suit called Montoya versus Bullock. I just want to share a little bit of history that Dr. Michael Trujillo, Miguel Trujillo's son, became the director of the Indian Health Service under the Clinton administration. He was the first American Indian graduate of the UNM School of Medicine. It's been my honor to work with Dr. Trujillo over the years. In his retirement, he served as the first director of our Cancer Center's Office of Community Engagement and Cancer Health Disparities. And he's been a trusted advisor for a long time. When we look today, there are 574 federally recognized sovereign tribal nations within the US territories. And they're divided by the Indian Health Service into 6 contract health service delivery areas that we call CHISTAs. And interestingly, you see that the Southwest CHISTA is a very large area, and that area is the subject for participant engagement for our grant. We also collaborate extensively with the Stevenson Cancer Center at the University of Oklahoma, who really is of service to the Indian nations in the Southern Plains, which you can see is a separate Indian Health Service delivery area. Now, what's interesting is tribal nations in the US, like all other racial and ethnic groups, are not monoliths. They have strikingly different genetic ancestries. One of the most interesting areas to me of science today is indigenous ancestral genomics and the tracing of indigenous populations across the world from early in time. And in the Southwest, we have 2 predominant ancestral lineages that I'll refer to later. The Puebloan lineage is quite distinct from the Athabascan lineage that derived both across the Bering Strait and came down from Asia into the US lands. And they're very different genetic lineages, even though those Indian communities are side by side. So throughout all of the 6 Chista regions, each tribal nation is highly diverse in genetic ancestry, in culture, in distinct languages and beliefs. But one unfortunate characteristic that these tribal nations share are tremendous cancer health disparities. There was a landmark study published in 2017 from the nation's cancer registries, published in the Journal of the National Cancer Institute by Jamal and colleagues. It was the 2017 Report to the Nation on Cancer, which actually focused on cancer survival. And this was the first time that it was demonstrated that American Indians and Alaskan Natives, compared to all other racial and ethnic groups in the US, are diagnosed at later stages of disease and have the poorest cancer outcomes for all cancers combined and for the most common cancers. This graph is lifted from that reference. And as you can see, it's mapping 5-year cause-specific survival. Shown in gold is the US average rate of survival in that cancer. And in the dark blue is shown the average American Indian cancer survival rate. So you see here, even for the very most common cancers, let alone disparities cancers, that American Indians have poorer outcomes for every type of disease. Returning to the Southwest Chista area, where we'll be engaging cancer patients and survivors, I want to talk a little bit about New Mexico and Arizona. So you again begin to see the incredible diversity of tribal communities in our region, which is a beautiful cultural richness. So in New Mexico, 11% of our population is American Indian, and it's composed of 19 Pueblo nations. If you look at the map of New Mexico on the left, you'll see all of those Pueblo nations noted in their location by a little blue dot. The Jicarilla and Mescalero Apache nations, which are separate Apache nations, and are also separate in that they are more of an Athabascan lineage related to Navajo than they are to Pueblo. In our northwest corner of New Mexico, you see the Eastern Agency of the Navajo Nation. And if you look carefully on the map, you see three separate Navajo bands, Rama, To'Hajiilee, and Alamo. And for those of you who know your history, Kit Carson forced the Navajo out of their native homelands in the mid-1800s to suffer something called the Long Walk to Bosque Redondo. And when they had to return after thousands died through starvation and disease, separate groups of Navajo broke off and didn't make it all the way back to their homeland and formed these separate Navajo communities. Albuquerque also has a very large urban Indian population, and many tribal languages are spoken. In the Pueblo communities, the predominant language are ancestral Puebloan, Tewa or Keres. The Navajo who speak their indigenous languages still speak an Athabascan dialect. When we look at Arizona, which Dr. Trent will talk about more, almost 5% of the Arizona population is American Indian, with 21 separate tribal nations that you can see on the map on the right. The largest nation, obviously, is the Navajo Nation in northeastern Arizona, which completely surrounds the Hopi Nation shown in red. There are also many ancestral Pueblo nations in Arizona, particularly the Salt River Pima Maricopa Indian community that TGen has collaborated with extensively, as well as Apache nations and the Havasupai and the Yavapai. One of the great benefits we have of studying cancer health disparities in the Southwest is the New Mexico Tumor Registry was formed in the early 1970s, and our state legislature mandated the reporting of all cancer cases statewide in this registry. So we have cancer incidence, mortality, disparity, stage data now since the early 1970s, which is quite remarkable. Our tumor registry also saved all the old tissue blocks. So we could go back and pull tissue blocks from some time ago and successfully isolate DNA or RNA or look at environmental contaminants using these blocks, which is allowing us to do really remarkable hypothesis-driven cohort studies. Now, interestingly, the New Mexico Tumor Registry also conducts population-based surveillance of all of the indigenous tribal communities in Arizona, as you can see on the map. So in the New Mexico Tumor Registry, we have data both from tribal communities in New Mexico and Arizona and can look at these trends. The New Mexico Tumor Registry is directed by Chuck Wiggins. It was a founding member of the NCI SEER program. I've mentioned it conducts population-based surveillance data in New Mexico and indigenous communities in Arizona. It has very detailed patient annotation, laboratory records, GPS coding of where patients reside, archived tissue samples, as I mentioned. And one of the uses we'll be using for the New Mexico Tumor Registry in our research center is they now, through the SEER program, can do rapid case ascertainment and identify for us very quickly newly diagnosed American Indian cases and their site of treatment or residence, allowing us to do rapid and early outreach to newly diagnosed patients in both of these states. One of the exciting things our registry just completed, also funded by the NCI Moonshot, was the development of our nation's first National Native American Indian Cancer Data website that integrates data from the NCI SEER, CDC, and Indian Health Service Tribal Epidemiology Centers. I've given you the link there. It's just opened in August, and it's quite remarkable for you to be able to go in and look at all Indian cancer data across all the Chista areas. I want to show you some of the data that is quite striking to me and to all of you, I think, that has led us to pick certain disparities cancers to focus on in our PE-CGS Center, as Dr. Carpton let you know. So this is a map, a figure of data from the New Mexico Tumor Registry that's reporting cancers of the colon and rectum in all New Mexico residents from 1983 to 2017. I think one of the most interesting thing about our registry data is we don't just look at a single point in time to see variation in racial and ethnic incidence. This is cancer incidence, but we can look at time trend changes, which are really striking and ask a lot of hypothesis-driven questions. So if you look at the first set of histograms, that's data averaged from 1983 through 1987, all the way to the right is the most recent data from 2013 to 2017. Gray, that gray bar on the right is US race incidence, all races combined. Dark blue, you see non-Hispanic whites in New Mexico. Sorry, that went, let me go back. Non-Hispanic whites in New Mexico, green are Hispanic, and gold is American Indian. And I think what you can see with colorectal cancer is while the incidence of colorectal cancer in the US and non-Hispanic whites in New Mexico has been reduced through better cancer screening programs, we're starting to see a decline in Hispanic. The rate has actually continued to increase in our American Indian communities. And we know they have some of the lowest rates of cancer screening in the United States. So this is a striking disparity where in New Mexico now, the highest rate of colorectal cancer is seen in American Indians. Next, I'll show you the data from cancers of the kidney and renal pelvis. And what you see is a striking increase, again, for the rate of kidney cancer in American Indians. American Indians in New Mexico have some of the highest rates of kidney cancer in the United States. And what's interesting about kidney cancer is if you look at all 6 CHISTA areas, you'll see this same data. And finally, this slide is showing the data in hepatocellular carcinoma. And again, Hispanics and American Indians have just dramatically high rates of hepatocellular carcinoma and intrahepatic tumors, even compared to the US population. And finally, I just want to show some data in breast cancer. We're going to be studying breast and hormone-dependent cancers as well, because again, you see that while compared to 1983 through 1987 and 2013 through 2017, the breast cancer incidence rates in non-Hispanic white women in New Mexico and the US population average haven't changed dramatically. They've actually increased a little bit. You see that there's been a significant increase in breast cancer in Hispanic and American Indian women. The genetic admixture of our populations haven't changed dramatically in this time. So again, we hypothesize this is due to dietary changes, exposural changes, and other changes that we're investigating. So what are the multi-level contextual factors that underlie these cancer health disparities? Well, I've told you that the rates of cancer incidence, mortality, and disparity vary among the CHISTA regions, but certain rates with certain CHISTA regions with higher smoking have higher lung cancer. That cancer is actually very low in the tribes in the Southwest region. But certain cancers like kidney cancer and liver cancer are high in all Indigenous communities in all CHISTA areas, which allow us to develop a lot of interesting hypotheses. The variable cancer patterns and disparities that we see obviously are multifactorial in causation, but we know one of the biggest for cancer such as colorectal cancer are significantly lower rates of cancer screening. And one of the challenges we see in the Southwest is screen positive patients are not rapidly navigated to care. So we have many intervention studies that I'll talk about in a few minutes at our cancer center, again, collaborating with Oklahoma and Arizona to try to enhance colorectal cancer screening and assure that screen positive patients are in fact navigated to care. We know there are different risk and protective behaviors among tribal communities in terms of cancer causation. Differences in diet, metabolism, obesity, tobacco, alcohol, and substance abuse. And we know there are highly variable rates of HPV and hepatitis virus vaccination. A very significant interest in our communities, which I'll talk a little bit about in the next slide, are environmental exposures from heavy metal mining waste, which is just endemic in the American Southwest. Plant toxins such as aristolochic acid or aflatoxin, particularly when our tribal communities grow their own corn, and other environmental contaminants. We assume and hypothesize that there are different spectra of cancer-promoting mutations, variable rates of inherited genetic alleles for cancer, but these have largely been unexplored. And another huge factor in disparities is just the limitations in immediate access to primary and tertiary healthcare, to cancer clinical trials, and access to modern cancer therapies, including precision medicine, immunotherapies, and targeted treatments. As I mentioned, environmental exposures is a significant concern of our tribal nations and leaders. These result from a legacy of abandoned hard rock mines in the West. If you look on the left, I've shown you the map of hard rock mines that are abandoned in the soft brown colors. On the right are the abandoned uranium mines and uranium waste sites. So greater than 50% of the tribal nations in the West in 15 states are located near or on greater than 160,000 abandoned hard rock mines and greater than 10,000 uranium waste sites. It's estimated now by the US EPA that over 40% of Western watersheds, air, ground, and soil are contaminated by heavy metals that leach into the water, into the air, and are admixed in the soil. The metals of concern that have been linked to cancer etiology include uranium, arsenic, lead, copper, cadmium, sorry everyone, and molybdenum and mercury. And so importantly, as I indicated, experimental and epidemiologic studies have linked these heavy metal exposures to increased cancer risk in both tribal and non-tribal populations. And there are two very important reports from the Strong Heart study. I've given you the reference there. The largest prospective epidemiologic study in American Indians, who are co-led by Dr. Jeffrey and Patricia Nez Henderson, who are collaborators at our center, that particularly found that exposure to cadmium and arsenic was linked to elevated cancer risk in American Indians. So what are we doing in our PE-CGS Center? We're focusing on cancer health disparities. We're going to focus on disparities cancers. And in the multifactorial context of all the factors that can promote disparities, as I mentioned, virtually unexplored are the genetic and genomic underlying mechanisms. If we look at American Indians and Alaska Natives, they've been strikingly understudied. Today, if you look at the Cancer Genome Atlas data, American Indians, Alaska Natives, and Hawaiian Pacific Islanders combined count for less than 0.5% of the patients studied. And to date, of the 11,000 patients in TCGA, only 27 are American Indian. So our hypothesis in our center is that through culturally appropriate collaborative participant and community engagement, respect of tribal sovereignty, cultural practices and beliefs, and comprehensive clinical grade genomic sequencing that's actually beneficent to a cancer patient because the data can be clinically used in their care, we'll discover novel somatic and germline mutations, differences in the spectrum or frequency of cancer promoting mutations in different cancers, and genome-wide mutational signatures that we believe could be translated to improve cancer screening, precision prevention, and therapeutic intervention in American Indian subjects. So our center has 6 goals to engage American Indian participants. We're going to be engaging actively newly diagnosed cancer patients and patients under our care, and survivors in tribal communities, in participating in research studies of the molecular characterization of cancers, focusing on cancers that display striking disparities in the Southwest. We hope to discover, as I just mentioned, novel somatic and germline mutations or difference in the spectrum and frequency of these mutations, and genome-wide mutational signatures reflective of behaviors and the kinds of environmental exposures I just mentioned. Scientists at the UNM Cancer Center have really recently identified mutational signatures that are unique for both arsenic and uranium exposure. We want to disseminate the knowledge that we gain through these studies to our tribal participants, who will be allowed the direct return of their clinical data for use at their request tribal councils, communities, and stakeholders through very unique means of participant engagement and novel means of communication using visual arts and media and Indigenous languages. I think it's important to realize that most Indigenous languages lack scientific and medical terms for words such as cancer, gene, DNA. So communication in Indigenous languages and using novel forms of engagement through communication are going to be essential. We'll share our knowledge with the broader scientific community through the NCI Genome Data Commons and the publication of results in accordance with NIH data sharing policies, which we've been allowed to modify, that they'll be dependent on tribal approval and tribal agreements. Ultimately, we hope, as I mentioned, to translate our discoveries to the development of targets and biomarkers that can enhance cancer screening and precision prevention, and to develop new diagnostics and therapeutics, thereby overcoming the tremendous cancer health disparities that I've shown you. We have a wonderful series of partners in our center. I will focus on two components of the grant, and Dr. Trent will begin to focus on the genome characterization unit. But we have built our team based on a strong history of deep engagement with Southwest tribal nations and communities. So I've already talked about the NCI SEER Tumor Registry, which we'll be using for rapid case ascertainment. We also have a prospective HPV registry and extensive research programs to enhance colorectal cancer screening in American Indian nations and communities in collaboration, a wonderful collaboration also funded by the NCI Moonshot between the University of New Mexico Comprehensive Cancer Center, the Stevenson's Cancer Center in Oklahoma, and the Arizona Cancer Center. Another thing we're building on are extensive programs with American Indian communities in environmental epidemiology, toxicology, and intervention research. I show you 3 grants listed here: metal exposure toxicity, a Center for Native American Health, Environmental Health Equity Research, and a new training program for Indigenous students in cancer genomics and environmental health. And finally, we have a statewide clinical trials network for patient engagement that involves virtually all of the community health systems in New Mexico who are going to be participants in this grant, consenting and submitting patients for the study. And that program is actually funded by an NCI Minority Underserved NCORE grant that will be pivotal for our work. Just to give you an example about what I mean about visual arts, we're currently doing one of the first chemo prevention studies in a tribal nation with the vitamin zinc. Scientists at UNM have found that uranium actually mediates DNA damage by tying up zinc finger binding proteins. And you can actually titrate out the uranium by oral zinc in animal models and get DNA repair. So to try to explain what Thinking Zinc, this interventional clinical trial is about, Mallory Kwataki from the Zuni Pueblo draws images like you see the uranium damages immune cell DNA, what the vitamin zinc does to sew that DNA back together, restoring healthy function, and can talk with individuals that were engaging in this chemo prevention trial in Navajo and Laguna Pueblo communities, actually using art and Indigenous language. We've also deeply engaged Dr. Trent and I, Dr. Johnny Lewis from the UNM Cancer Center with the NCI, both in the Southwestern tribal nations and the South Central Plains, in several meetings to engage tribal leaders to lay the groundwork to be able to do these studies. In 2016, we worked with the NCI to do the first Precision Medicine and Cancer National Conference, which we held at the Stevenson Cancer Center. And in 2017, we actually hosted the Navajo Nation and the NIH Tribal Health Research Office that led to the large now NIH Tribal Data Sharing Agreement. And in 2019 and 2020, we've hosted many meetings, TGen and UNM, both here in New Mexico and Arizona, to engage tribal leaders in the passing of formal resolutions to allow us to develop collaborative genomic studies with their communities and populations. Shown on the right is a historic resolution we're very proud of, working with the All Pueblo Council of Governors that outlines their intent to work with us to develop a formal biomedical consortium legal construct, similar to what Dr. Trent will talk about with the Salt River Pima in Arizona, to conduct interventional studies, cancer genomic studies, and environmental studies in collaboration with tribal nations. I'm very honored to have this resolution in support of the El Pueblo Council of Governors. Just a few words about our units, and I'll turn this over to Dr. Trent. So one of our wonderful partners in our center are not only about 40 investigators from the University of New Mexico Comprehensive Cancer Center, but also investigators from the Black Hills Center for American Indian Health. So the participant engagement unit is based in New Mexico at UNM, and we have a cancer patient engagement team led by Dr. Rich Lauer, a community participant engagement team led by Dr. Andrew Sussman. As you can see in both cases, we have both Indigenous speaking tribal members who will be working on these engagement activities. One of the important things we're going to be doing is collecting epidemiologic and behavioral data on all of our patients as well. That's something missing from TCGA today. That's inhibited our ability to do the kind of data correlatives we want to do. Our PE-CGS engagement optimization unit is led by Dr. Shiraz Mishra, and I want to mention Dr. Stacy Gray from City of Hope, who's an expert in using automated e-health means to return results directly to patients. And we're really thrilled to have Stacy and Gray engaged in the project. Both Dr. Jeffrey Henderson, who is Cheyenne River Sioux, and Patricia Nez Henderson, who's Navajo, who formed the Black Hills Center for American Indian Health, will be working with us. We're thrilled to have them. They've led the Strong Heart study and many other large, both population-based tobacco cessation and genetic studies in American Indian nations, and actually have an office in Window Rock, Arizona on the Navajo Nation, and will be assisting and advising us to interface with the Navajo Nation for the project. And finally, RS21, Resilient Solutions for the 21st Century, is a wonderful IT AI firm in Albuquerque that will be building a very large integrated patient data warehouse where we can tie the genomic data to therapeutic intervention data, the epidemiologic and behavioral data, and all of the patient annotation data in the project. We plan to study 1,000 American Indian cancer patients and survivors with disparities cancers during the 5 years of the grant. And as I've shown you in the table on the right, we've carefully looked at the number of cases we believe we can ascertain in each year in gastrointestinal and hepatobiliary cancers, genitourinary cancers, and hormone-dependent cancers. So we plan to engage about 152 patients per year that we think will consent to this study in these disparities cancers who are active cancer patients, and another group of survivors that together total about 1,000 patients over a 5-year period. I'll mention that here at the Cancer Center at UNM, we see about 220 newly diagnosed cancer patients a year. Half of those patients roughly are Pueblo, half are Navajo and Apache. Through the generous support of a donor, we'll also be sequencing 1,000 Hispanic patients with these cancers in a matched cohort. As Dr. Trent will describe, we'll be using CLIA-grade comprehensive sequencing through their company, Cheyenne, and we'll be returning results directly to patients and providers in the context of clinical genetic counseling. And finally, to close, I just want to touch base again on the sensitivities of collaborating with tribal nations from a respectful position, acknowledging that they are sovereign tribal nations. And there are a lot of sensitivities, as many of you know, with justification from these tribal nations. Through many research studies historically, their tribal sovereignty has not been honored. Certainly we do that and have worked closely to develop formal consortium and collaborative agreements to enable us to do these studies. The tribal councils will be approving IRB for all of our studies. The first IRB approval has been obtained and have been reviewing the actual processes we'll use for informed consent. We plan to have a monitoring committee that looks at joint biospecimen use. Participants will be able to consent to not have any of their specimens stored, or if such specimens are stored, they have to be reconsidered or not for any future use. There's a great deal of sensitivity for biospecimen use in tribal nations, much like working with traditional Jewish communities or other racial and ethnic groups who have these sensitivities. While we will be submitting data to the NCI Genomic Data Commons in real time, that data will be held for public access until we work with tribal communities to develop the interpretive data narratives and actually publish the work. Future use of that data will require tribal approval, particularly any studies that might explore ancestry or ancestral linkages. And then finally, I want to close my comments by acknowledging the amazing tribal leaders I've worked with, the All Pueblo Council of Governors, prior Chairman Paul Torres, and now Chairman Michael Chavarria, who's the governor of the Santa Clara Pueblo, the Albuquerque Area Southwest Tribal Epidemiology Center, Dr. Kevin English, Jonathan Nez, president of the Navajo Nation, and several tribal leaders and advisors, Dr. Regis Pecos, Dr. Kurt Riley, Eric Lujan, Gregory Ortiz, and Dr. Michelle Swina, who've given us wonderful advice and support over the last 20 years. So thank you very much.
Thank you, Cheryl. That's great. Jeff, if you can unmute your mic.
I will.
See your slides fine.
Okay, so thank you very much. Thanks, Dr. Carpenter, for your kind introduction, Cheryl, for the outstanding introduction to the project. Again, I'm a professor at TGen at the City of Hope Cancer Center, and I'm going to focus really very briefly on three major areas. One, as described by Dr. Willman, just that sense of the clear importance of both building and maintaining trust within tribal communities. So I'll briefly mention a nearly 2-decade-long partnership between TGen and the Salt River community. I'll talk about what we're going to be mapping in regards to the molecular basis of cancer through this project. And finally, some of the challenges and the strategies for improving variant calling that's so important because we are attempting to deliver information back for patient benefit. It's important to point out that this project is part of what is a new and evolving moonshot network, this PE-CGS, one that's managed across the NCI by Drs. Mechanic and Glanders and others. But it has as its core an expectation that we will deliver clinically relevant information, not simply to a database, but back to the benefit of the study participants. Focusing very briefly on the Salt River Pima Maricopa community, as mentioned, there are 21 communities. The Salt River Pima Maricopa Indian community sits next to Arizona's largest city, Phoenix. If one were to go back and look when we began this discussion with TGen and the community members, this is the Arizona Cancer Registry Health Services data over the period 2005 to 2009. And it shares data for cancer incidence across the tribal communities, all of the 21. And the Salt River rate for all cancers, as you can see, is almost 1.7-fold higher. In fact, the highest rate of all cancers for all Arizona Indian communities. Working with the community leadership, particularly Violet Michelinos and the council and president at that time, they really reached out to us to said, we have to solve the issue that researchers are not just those that parachute in. You have to spend enough time to work with us. These are the improved areas that the community asked for. So they asked us to closely work with the communities to develop these programs, the needs of the community, and that literally they would decide what we would work on, not we would tell the community what we would work on. So that and providing communication was a recommendation, as well as truly establishing a partnership and a model for that would include everything, including data ownership. So the research agreement study history for TGen and Salt River Pima is characterized on this slide. Lots of interaction with the medical director, health staff, and really they identified this, what you heard from Dr. Willman, the significant number of patients diagnosed with renal cell carcinoma, and they picked that as a joint study development component. I'll point out that, again, this research plan, and remember, this was established in 2007, the picture shown on the right with the president at that time, President Diane Enos, and myself signing this agreement 13 years ago, included MTAs, intellectual property agreements, publication policies, IRB, and then what a real blessing to a number of us was our laboratory areas and staff blessed by tribal elders as well. We've continued regular contact with the Department of Health and Human Services, current director, Joseph Rimanetra, as well as the tribal president who actually sits on our non-fiduciary board of governors at TGen, and I give quarterly reports to the tribal council. This is a major issue, as you heard from Dr. Willman. Average age of diagnosis for individuals in this community at 56 years of age, if you look past over, is 10 years earlier than the national average. And if you look at the death rate to the disease, so as you know, not everyone who gets cancer dies of it. This death rate is 3 times the US national average. And then finally, if you say what contribution has TCGA, this is not a negative, this is just data. There are 0 patients that have been published across the 2 cancer studies in renal cell. Actually, there's one, a Alaskan native, but 0 Southwest American Indians were included in that study. And again, we're very excited to be able to suggest that even in the small cohort we've been able to look at, which will be scaled tremendously through this study and our work with Dr. Willman, that we'll be able to, we believe, identify unique genetic signatures that are community specific, useful for diagnostic, screening, and treatment relevance. Now, very quickly, I'm going to mention to you again, and forgive me for this detail, many of you will be quite familiar with this type of precision medicine workflow that begins with Dr. Lauer and his group at New Mexico that brings together the patients, the consents, and the other aspects, and across many very talented investigators. And at the heart of it is the expectation that we must be able to deliver return of information. So therefore, at the center has to be the ability to perform CAP-CLIA-based sequencing. So it's clinical collaborations all the way through data, tumor boards and data sharing, which includes, for those of us that work in this area, we recognize a massive amount of information and a massive number of individuals that will be engaged and are engaged as we begin to bring this into reality. Now, I'm only going to mention that the work that we do is inside our laboratory. It's our CAP-CLIA lab, totally under the nonprofit TGen, which is part of City of Hope. And our exome transcriptome that we do is based on a little over 19,000 genes, 800x average coverage, well, 300x over all the genome, 800x over about 440 genes. And ultimately, if you look across the last 1,000 or so, we've had more than 950 average X coverage for cancer and about 450 across the rest of the genome. We do RNA sequencing, that's ribodepleted RNA, and this is tumor normal. It's also just recently been approved with national coverage determination by CMS. Now, the part I'm going to raise just very briefly is that this didn't happen by accident. It happened because there was a remarkable effort to establish and work with groups that are establishing genomic standards. And I'll return to this in the last part of my talk. Multiple manuscripts published at TGen from the multiple faculty that sit and work on aspects for reference standards and quality control for RNA and DNA. And I took this opportunity to show the picture of Dr. Carpton at the time that he was at TGen, along with Dr. David Craig, the both the professors at USC now that did one of the first somatic standard reference genome sequencings put into the public domain in this scientific report 2016 that gave us an opportunity to utilize it for the first time with the oversight of the FDA and device pre-submission that allowed us to begin to apply this not just in clinical trials, but ultimately through now CMS approval as well. Well, with all that as a background and trying to just leverage the idea that we have to be able to deliver clinical information back, what is the genome characterization going to capture? Because this will be data of tremendous use for the overall community. So in CLIA, DNA and RNA extraction held in CLIA chain of custody, tumor normal exome sequencing, RNA-seq, which allows you to do all the aspects that are critical for being able to provide information back from RNA fusions to amplifications, deletions, TMB, et cetera. Also, we will be doing whole genome sequencing at approximately 30x under RUO, which will allow us to capture, just as you heard from Dr. Willman, mutation signatures, work that's been done with Ludmila Alexandrov and others, as well as circulating tumor DNA. And for about 200 of the 1,000, we plan to be able to also incorporate microbiome, both for saliva and for stool. Now, I'm not going to be able to show data because we've just started this project, but I will say that if you begin to integrate this data, it makes a huge difference. And an example is work at TGen, Jonathan Keats and his group looking at the MMRF study that, again, Dr. Carpton was involved in during his time at TGen as well. An observational clinical trial, 8 years of observation, data collected every 3 months, multinational clinical information. But if you looked at the integration of this, the integration of 3 data sets and sources, 5 different tumor types, these are of data types, again, very similar to what we're going to capture, long insert, shallow genome, exome capture at high coverage, RNA-seq to allow us to really look across structural alterations, rearrangements, mutations, LOH, et cetera. 982 patients looking at this. Now, why did I show this? And that's because absolutely biological insights from this type of integrated approach can change clinical interpretation. And this is an example for looking at 3-way analysis of TP53 mutation within this subset. And everyone that's shown on the left in the high risk category for myeloma would have been considered and offered aggressive therapy. We now know on the right that that if you integrate this data, that the mutations are mostly found when a deletion exists, and when you separate patients, we either have 2 copy loss, complete loss, 1 copy loss, no allele loss. Only poor survival is associated with those with complete loss. So we would have been, and we are giving patients bad information today without this type of information. And we already know this from Dr. Willman's work in leukemia. We're going to find these type of extraordinarily important components going forward. I will only mention that we are doing circulating tumor DNA. We have unique ways to look across the tumor portals to share data. But I do want to just very briefly mention that the challenge and strategies for improving variant calling are critical. Improving the success rate for precision medicine is absolutely central. And part of that is that one human genome assembly does not fit all population. Without going into major discussion, this is an area that Dr. Carpton is also an expert in. The background problem is that personalized medicine is not particularly personalized unless you happen to be of an American that has Northern European ancestry. So in order to reliably identify this, part of the solution, it's just part of the solution, would be to develop an American Indian specific reference human genome, something that we're working on using de novo assembly, and I'll just mention it in the briefest way. With the remarkable work of Nick Shork and a whole team of investigators, we've been benchmarking phasing strategies is one example of why this is so important. And I'll simply say that using just standard efforts across published data, for example, from the 1,000 genome projects, the switch error rate, this is really what's characterized on those two slides on the panels on the right. Just point out the fact that we have to be able to increase both the sample size, reference panel, and minimal switch erroring rates, because the reality is that we have to be able to use this for personalized medicine. Now, this is not a unique issue. I mean, there are wonderful projects, 1,000 genome, haplotype reference, TopMed. But if you really say in the existing genome assemblies and variant databases and available annotations, how much information is there on American Indian communities? And that answer is almost none. So again, this is a critical area. Now, what our first step for this is to begin building human reference genomes for American Indian communities. And I'll simply say, again, that we're doing this now. This used to be a project that you needed to address at the national level. We're addressing it because of the leveraging of advances in computing and security that allow us to do de novo assembly using GPUs instead of CPUs, and maximizing and reducing time that we think are going to be very important as described on this slide. Now, I realize that time is certainly very short. I appreciate the opportunity to have presented some of the challenges and what we're working on. And now I'll turn this back over, and hopefully there'll be time for discussion for either Dr. Willman or myself. Thank you very much.
Outstanding, and really exciting, and will provide tremendous amount of data to close many really important gaps. And as you mentioned, we don't have a lot of time, but there are a number of questions and comments, and we'll make sure to share these with the speakers in order for them to sort of get a handle on what types of things the community I might be thinking about, in reference to your program. And Nicole Roberge, made a couple of interesting comments, not necessarily questions, and mainly about sort of the tribal sensitivities and research and in Native Americans or American Indians. And it's interesting, she mentioned a scenario by where a researcher was presenting at a conference and didn't realize that the Navajo IRB member was in the audience, and called the researcher out for failing to respect the people in obtaining IRB approval. And we know about these sensitivities. So there, of course, were a number of questions about that. For instance, Dr. Galanders asked the question, how will the Navajo Nation moratorium on genetic research studies impact your study? And I don't know if Jeff or Cheryl, maybe you can both unmute your mics and as we walk through the questions and comments.
So I'll take a first crack at that and let Jeff pick up. As we have met with the Navajo Nation and talked about these studies, our work in the formation of our formal biomedical consortium agreement has primarily engaged the Navajo Nation, the Pueblo Nations in New Mexico. We were really launching that work in March when the COVID epidemic hit. And as many tribal communities were closed, unfortunately, we've had to set that work aside. We hope to just begin getting started again. And our tribal leaders of the All Pueblo Council of Governors and the Health Subcommittee, who we're working with, and their attorneys plan to invite the Navajo Nation to engage with us in those discussions. In general, the attitude of our patients and the Navajo Nation has been, for an individual cancer patient, there is support for them selecting genomic studies for their individual use if they're beneficent and benefit that patient. So we actually have many Navajo patients in our cancer center who elect to participate in precision medicine studies for their personal use. Studies of survivors in the community, I think, will take much deeper scrutiny of the Navajo Nation. So we plan to just stage this, John, as we go forward. We're very respectful of the moratorium on genetic sequencing. There's been a lot of recent discussion of the Navajo Nation, of the South Central Research Foundation in Alaska, about beginning to allow genetic studies for clinical use.
Right. So again, there are a ton of questions, and boy, I think they're all great questions, and I wish we could get through them, but I know we're running out of time. But just one last sort of, I think, broader concept around research in cancer disparities, and of course with the very unique aspects related to American Indians. So one question was about the rate of participation in clinical trials among Native American populations, and a related question: can you talk about the specific barriers to enrollment in clinical trials and biomedical research that you've encountered, and some of the strategies that you've developed to mitigate or overcome those barriers?
So again, as we're short on time, I'll answer quickly. A couple questions I see. I want to emphasize again, as Dr. Trent did, that yes, individual participants who consent to the study do receive their own data. They'll be receiving formal reports of their sequencing analysis in the context of a clinic setting with a genetic counselor who can explain that data to them and any other selected family members that they choose. So we will be returning results if that is requested by the patient, and we anticipate most patients will want access to their own data just based on our experience to date. And then, John, speaking about clinical trials, I think there are many challenges. I mean, if you look at American Indian accrual to national clinical trials in the US, it's abysmal to non-existent. A lot of that, I think, is clinical trials aren't accessible to Native American participants. A huge factor we've been working on at NCI is that the comorbidities often exclude patients because the eligibility criteria for so many clinical trials are just too restrictive. So there have been huge projects at NCI we really need to drive home about looking at eligibility criteria and not excluding so many patients due to comorbidities. And then finally, I think when we look at many of the disparities cancers that Dr. Trent and I plan to study, there aren't huge clinical trial menus in those cancers. And so it's really important to begin trial menus that match the disparities cancers that we see not only in American Indian, but in Black, Hispanic, and Asian subjects across the United States. We've had an unconscious bias towards developing clinical trials for non-Hispanic white people. That has to change.
Sure. Well, thank you both so much. Jeff, I don't know if you had any closing comments but all the questions are saved. We'll make sure that the questions get to our speakers, our panelists. I know, for one, I'm really excited about this study and can already begin to envision, the tremendous impact this will have on building models, broader models that can be distributed more, across cancer centers to better engage and involve underrepresented minority patients and the medically underserved into biomedical research studies. So we thank you both for that tremendous presentation.
Great.
Yep. Thank you, everyone, for a great presentation today and your participation in this webinar, and the moderation and the Q&A was great. I want to remind you that our next Cancer Moonshot webinar is Thursday, November 19th. This is Dr. Charles Sawyers talking about developing ways to overcome cancer's resistance to therapy. You can register now on the Cancer Moonshot webinar page. And thank you, everyone, for your participation. I hope you have a great day. Thanks.
Thank you.